This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome. Ever wonder how microbes, the tiniest life forms, thrive in competitive environments? This exploration of the Black Queen hypothesis reveals a surprising evolutionary strategy. Sometimes, losing is the key to winning. In easy-to-understand language, we'll uncover how shedding genes can be advantageous, exploring the power of microbial communities, the risks and rewards of interdependence, and the delicate balance between cooperation and exploitation. Get ready to discover a fascinating new perspective on evolution and the surprising ways microbes interact. Chapter 1. The Queen's Gambit. Why losing is the new winning in microbial evolution. Forget the gladiatorial arena. Evolution isn't always about a brutal fight to the death, a constant one-upmanship of ever-increasing complexity. Sometimes, the most successful strategy involves a different kind of gambit, a clever maneuver where shedding, rather than acquiring, becomes the key to thriving. This is the surprising wisdom of the Black Queen hypothesis, a theory that reveals how losing genes, like discarding a burdensome card in a high-stakes game, can be the winning hand in the microbial world. Picture the open ocean, a seemingly limitless desert of water where life teems in microscopic abundance. Resources are precious in this vast expanse, and competition is a constant undercurrent. Yet, paradoxically, some of the most dominant organisms in this harsh environment are those with the smallest genomes, streamlined down to the barest essentials. These microbial minimalists, like the incredibly abundant Prochlorococcus and Pelagibacter, have taken the evolutionary gamble of discarding genes that appear, at first glance, absolutely crucial for survival. Pelagibacter, another incredibly abundant ocean dweller, has a remarkably streamlined genome, lacking the genes to create certain essential nutrients. It's as if they've outsourced their grocery shopping, relying on others to stock the shelves with the necessities they can no longer produce themselves. So how do these seemingly vulnerable microbes not only survive but flourish in the competitive ocean environment? The answer lies in the power of community. The Black Queen hypothesis proposes that these lost functions aren't truly gone. They've simply been outsourced to other microbes in the community. This creates a web of interdependence, where some microbes thrive by relying on others. The hypothesis borrows its name from the card game Hearts. For those unfamiliar, the goal of Hearts is to finish the game with the fewest points. Points are accumulated by taking certain cards, most notably the Queen of Spades, which carries a heavy penalty. Skilled Hearts players strive to avoid taking the Queen of Spades, even if it means sacrificing other cards. This strategic avoidance mirrors the evolutionary gamble taken by microbes like Prochlorococcus and Pelagibacter. The genes they've lost are like the Queen of Spades, costly to maintain but ultimately dispensable if someone else is shouldering the burden. By shedding these genetic responsibilities, the beneficiaries free up precious resources that can be redirected towards growth and reproduction, giving them a competitive edge in the resource-limited ocean environment. But like any gamble, there are risks involved. Losing essential genes is a high-stakes maneuver. What happens if the helper population declines, or worse, disappears entirely? The beneficiaries, now stripped of their safety net, become vulnerable to the environmental stresses they once could handle themselves. The gamble can lead to a catastrophic loss. This inherent risk is a driving force in shaping the intricate interconnectedness of microbial communities. It creates a delicate balance between dependence and independence, where the fate of one species is inextricably linked to the fortunes of others. The Black Queen hypothesis offers a fascinating new perspective on evolution, challenging the traditional narrative of ever-increasing complexity. It reveals a hidden layer of cooperation and interdependence in the microbial world, where losing, sometimes, is the most ingenious way to win. It forces us to rethink the concept of individual success, recognizing that in the microscopic realm, community matters more than we might have ever imagined. This realization has far-reaching implications, not only for our understanding of microbial ecology but potentially for other biological systems, even extending to the complex web of interactions within human societies. Perhaps, the Queen's Gambit is a more common strategy for success than we realize. Chapter 2. Leaking secrets. The unexpected benefits of imperfect biology. The microbial world thrives on a principle that seems counterintuitive: imperfection. In this bustling metropolis of microscopic life, leaky functions, biological processes that inadvertently spill their benefits beyond the individual organism, are not flaws, but the very foundation of community and cooperation. These leaks create a shared resource pool, a commons that fosters interdependence and drives the evolution of intricate relationships between diverse microbial species. It's a world where giving, even unintentionally, becomes a powerful strategy for success. Imagine a microbe diligently breaking down a complex sugar molecule, like cellulose, for food. This process, mediated by specialized enzymes, releases smaller, more digestible sugar molecules. Inevitably, some of these sweet treats escape the cell's grasp, diffusing into the surrounding environment. What appears as a loss for the individual becomes a boon for its neighbors, a readily available food source for those lacking the enzymatic machinery to break down cellulose themselves. This leakiness is not a design flaw, but a fundamental aspect of biological systems, creating a web of interdependence where one organism's waste becomes another's treasure. This principle extends far beyond the simple sharing of sugars. Leaky functions encompass a diverse array of biological processes, each contributing to the microbial commons in unique ways. Consider the detoxification of harmful substances. In the sunlit surface waters of the ocean, hydrogen peroxide, a reactive oxygen species, poses a constant threat to microbial life. Some microbes, like certain strains of Synechococcus, produce catalase peroxidase, an enzyme that neutralizes hydrogen peroxide, protecting themselves and inadvertently shielding their neighbors from this toxic chemical. This shared defense allows the more sensitive species to thrive in an otherwise hostile environment, highlighting the protective shield created by leaky functions. The costs associated with these leaky functions vary depending on the specific function and the environment. Producing an enzyme like catalase peroxidase requires a significant investment of resources, particularly iron, which can be scarce in the open ocean. The balance between the cost of producing a leaky function and the benefit received can influence the evolution of dependencies. In environments where a leaky function is readily available, the cost of producing it oneself might outweigh the benefit, creating a selective advantage for organisms that shed this burden and rely on the commons. This is the heart of the Black Queen hypothesis, which explains how leaky functions create the conditions for gene loss and the evolution of dependencies. By relying on others for these shared resources, microbes can streamline their genomes, discarding costly genes and freeing up resources for growth and reproduction. This creates a delicate balance of interdependence, where each microbe contributes to the shared resource pool while benefiting from the contributions of others. It's a microbial menagerie of cooperation and competition, where leaky functions are the currency of exchange. It's a story of imperfection leading to unexpected benefits, a testament to the power of community in the microbial world. Chapter 3. A Microbial Menagerie: Cooperation, Cheating, and the Tragedy of the Commons. The microbial world, a bustling metropolis of microscopic life, thrives on a delicate balance of cooperation and competition. While the previous chapter illuminated the surprising benefits of leaky functions and the evolution of interdependence, this chapter delves into the darker side of shared resources, the potential for exploitation and the microbial equivalent of the tragedy of the commons. The tragedy of the commons, a concept borrowed from human economics, describes a scenario where shared resources are overexploited and ultimately depleted due to individual self-interest. This same principle can play out in the microbial world, where public goods, the beneficial molecules leaked into the environment, can be exploited by cheaters, microbes that benefit from the shared resource without contributing their fair share. In the context of the Black Queen hypothesis, cheaters are microbes that have lost the genes for a leaky function but still benefit from the public good provided by others. This genetic streamlining allows them to reproduce faster, outcompeting their more generous counterparts. While the loss of a single cheater's contribution might be negligible, the proliferation of cheaters can dramatically increase the demand on the shared resource, potentially pushing the system towards a tragedy of the commons. Consider the production of siderophores, iron scavenging molecules secreted by many microbes. Iron, essential for numerous biological processes, is often scarce in the environment. Siderophores act like molecular magnets, binding to iron and making it available for uptake by microbial cells. However, siderophore production is energetically costly. Cheaters, having lost the genes for siderophore synthesis, can still acquire iron by snatching up the siderophores produced by others, gaining a competitive advantage without bearing the production burden. As cheaters become more abundant, the demand for iron increases, while the supply of siderophores, limited by the dwindling helper population, decreases. This imbalance can lead to iron scarcity, hindering the growth of the entire community, including the cheaters themselves. A similar scenario can unfold with the secretion of extracellular enzymes, another crucial public good in many microbial communities. These enzymes, released into the environment, break down complex organic molecules, such as cellulose or proteins, into smaller, more digestible units. This breakdown process benefits the entire community, providing a shared pool of readily available nutrients. However, enzyme production is an investment. Cheaters, lacking the genes for enzyme synthesis, can still access the breakdown products, effectively free riding on the efforts of the enzyme producers. If cheaters become too numerous, the supply of breakdown products might not keep pace with the demand, leading to nutrient scarcity and impacting the entire community. The Black Queen dynamic itself can contribute to a tragedy of the common scenario. As beneficiaries become increasingly reliant on helpers, the community becomes vulnerable to fluctuations in helper populations. A decline in helper numbers due to environmental changes or the invasion of a new predator can have cascading effects on the entire community, leading to a shortage of public goods and jeopardizing the survival of the beneficiaries. Similarly, the emergence of highly efficient cheaters, those that are particularly adept at exploiting the public good without contributing, can destabilize the established equilibrium, potentially driving the helper population to extinction and triggering a collapse of the shared resource. However, microbial communities are not passive victims of the tragedy of the commons. They have evolved a variety of mechanisms to mitigate this risk and maintain a balance between cooperation and cheating. Spatial structuring is one such mechanism. By forming biofilms or aggregates, microbes create microenvironments where public goods are concentrated, primarily benefiting the producers and their close relatives. In soil, for instance, microbes often cluster within microaggregates, small clumps of soil particles bound together by organic matter. These microaggregates create protected pockets where secreted enzymes and their breakdown products are less likely to diffuse away, favoring the producers and limiting access for cheaters. Similarly, in biofilms, the intricate architecture of the community can create gradients of public goods, with higher concentrations near the producers and lower concentrations further away. This spatial heterogeneity can favor producers and discourage cheaters, promoting cooperation and stability. Different biofilm structures, such as mushroom-shaped structures or densely packed layers, can further influence the distribution of public goods and the interactions between producers and cheaters. Frequency-dependent selection is another powerful mechanism that maintains balance. In this scenario, the fitness of a particular genotype depends on its prevalence in the population. Consider a microbial community where some members produce a costly antibiotic to defend against a common competitor. When cheaters, who don't produce the antibiotic, are rare, they thrive, benefiting from the protection provided by the antibiotic producers without bearing the production cost. However, as cheaters become more abundant, the competitor, no longer effectively suppressed, rebounds, impacting the entire community, including the cheaters. This resurgence of the competitor gives an advantage back to the antibiotic producers, whose numbers can then recover. This dynamic interplay between helpers and cheaters creates an oscillating equilibrium, preventing the complete dominance of either genotype and promoting the long-term stability of the community. Quorum sensing, a regulatory mechanism used by many microbes, can also play a role in managing public goods and potentially preventing overexploitation. Quorum sensing allows microbes to communicate with each other by releasing and detecting signaling molecules called autoinducers. As the microbial population grows, the concentration of autoinducers increases, triggering a coordinated response, such as the production of a public good. This density-dependent regulation can prevent the wasteful overproduction of public goods when the population is small and the benefit is minimal. The Black Queen dynamic can influence quorum sensing in various ways. For example, beneficiaries might evolve to be more sensitive to autoinducers, allowing them to respond more efficiently to the presence of helpers and their public goods. Conversely, helpers might evolve to produce autoinducers more efficiently or to release public goods only when autoinducer concentrations reach a certain threshold, ensuring that the public good is primarily available to producers and their close relatives. The diversity of leaky functions and public goods in different microbial environments is vast. In the ocean, the degradation of dimethylsulfoniopropionate, DMSP, a compound produced by phytoplankton, releases dimethyl sulfide, DMS, a volatile gas that contributes to cloud formation. This process, a leaky function, influences global climate patterns. In the human gut, microbes produce short-chain fatty acids, such as butyrate, which provide energy to the gut lining and have anti-inflammatory effects. These fatty acids, a public good, contribute to human health. In soil, microbes produce a variety of antibiotics, some of which have been harnessed for human medicine. These antibiotics, a leaky function, can shape the composition of the soil microbial community and provide protection against pathogens. The Black Queen hypothesis has profound implications for understanding microbial community stability and resilience. The loss of a key helper population, for instance, can destabilize the entire community, leading to a shortage of essential public goods and potentially triggering a cascade of extinctions. Similarly, the emergence of new cheaters can disrupt the established equilibrium, potentially leading to a tragedy of the common scenario. Understanding these dynamics is crucial for predicting how microbial communities will respond to environmental changes or disturbances, such as pollution, climate change, or the introduction of new species. The microbial world is a complex and dynamic ecosystem, where cooperation and cheating are intertwined. The Black Queen hypothesis provides a framework for understanding this intricate interplay, highlighting the importance of leaky functions, public goods, and the evolution of dependencies. It's a story of give and take, where the fate of the community depends on the delicate balance between individual selfishness and collective responsibility. Chapter 4. The Microbial Marketplace, a Network of Dependencies. The microbial world isn't a simple dichotomy of helpers and freeloaders. It's a bustling marketplace, a complex network of dependencies where microbes barter and trade essential functions, creating a dynamic spectrum of interactions that ranges from mutually beneficial partnerships to outright exploitation. The Black Queen hypothesis, far from depicting a static landscape, provides a framework for understanding how this microbial marketplace operates, how dependencies evolve, and how the balance of power can shift dramatically in response to environmental pressures and evolutionary innovations. Consider, for instance, the cooperative exchange between certain soil bacteria and fungi in the rhizosphere, the nutrient-rich zone surrounding plant roots. Bacteria, equipped with the genetic machinery to synthesize essential amino acids, leak these vital building blocks into the environment. Fungi, in turn, excel at breaking down complex plant polymers, releasing simple sugars that become a readily available food source for the bacteria. This mutually beneficial exchange, a classic example of mutualism, demonstrates how leaky functions can create a stable partnership, where each partner contributes a unique and valuable service to the other. This isn't simply a one-way street of bacterial generosity. Fungi also leak some of the sugars they produce, creating a feedback loop of reciprocal benefit. This exchange is further influenced by the plant itself, which releases root exudates, a cocktail of sugars, amino acids, and other organic compounds, shaping the rhizosphere environment and influencing the interactions between bacteria and fungi. However, this seemingly harmonious marketplace can quickly turn cutthroat. Imagine a scenario where a new bacterial species arrives, one capable of synthesizing its own amino acids and scavenging the sugars leaked by the fungi. This newcomer, a microbial entrepreneur, disrupts the established equilibrium. The original bacteria, now facing competition for sugars and no longer holding a monopoly on amino acid production, might experience a decline in population. The fungi, on the other hand, could benefit from the increased competition for their leaked sugars, potentially driving the evolution of more efficient sugar production or transport mechanisms. This example illustrates how the arrival of a new species, or even a change in environmental conditions, can trigger a cascade of changes, reshaping the network of dependencies and altering the balance of power within the microbial marketplace. The Black Queen hypothesis can also lead to more complex, multi-layered dependencies, where a single species can be both a helper and a beneficiary for different functions. Consider a microbial community where one species, let's call it species A, detoxifies hydrogen peroxide, a leaky function that benefits the entire community. However, species A is oxytrophic for a specific amino acid, relying on another species, species B, for its supply. Species B, in turn, might depend on a third species, species C, for the breakdown of a complex carbohydrate. This creates a chain of dependencies, where the fate of each species is linked to the others. If species C disappears, species B might struggle to survive, impacting the supply of amino acids and ultimately jeopardizing the survival of species A. Even though species A doesn't directly interact with species C, these indirect effects highlight the interconnectedness of the microbial marketplace and the potential for cascading failures when a key player is removed. This interconnectedness can also lead to the evolution of keystone species, organisms whose rarity belies their crucial role in maintaining community stability. Imagine a microbial community in a deep-sea hydrothermal vent, an environment rich in sulfur compounds. One rare species, a microbial specialist, might possess the unique ability to oxidize sulfide, a toxic compound, converting it into sulfate, a usable form of sulfur for other community members. This sulfide oxidation, a leaky function, creates a habitable zone around the specialist, allowing other species to thrive. Despite its low abundance, the specialist plays a crucial role in maintaining the entire community. Its loss would lead to a buildup of sulfide, potentially poisoning the surrounding environment and triggering a cascade of extinctions. This example illustrates how a helper organism, by providing a crucial public good, can become a keystone species, its presence essential for the survival of the entire community. The rarity of keystone species makes them particularly vulnerable to environmental changes or disturbances, highlighting the fragility of microbial ecosystems and the importance of preserving biodiversity. Environmental factors play a crucial role in shaping the microbial marketplace and the types of interactions that arise under the Black Queen hypothesis. Resource availability, for instance, can influence the balance between cooperation and exploitation. In nutrient-rich environments, where resources are abundant, the cost of producing leaky functions might be low, favoring cooperation and the evolution of mutualistic relationships. However, in nutrient-poor environments, where resources are scarce, competition intensifies, and the selective pressure to shed costly functions and become a beneficiary becomes stronger. This can lead to an increase in cheating and a shift towards more exploitative interactions. The presence of other species can also influence the evolution of dependencies. In a diverse community, where multiple species perform similar leaky functions, the selective pressure to lose a particular function might be higher, as beneficiaries have multiple potential helpers to rely on. However, in a less diverse community, where a single species is the sole provider of a crucial public good, the loss of that species can have devastating consequences. This highlights the importance of biodiversity in maintaining community stability and resilience. The loss of biodiversity can disrupt the intricate web of dependencies, making the community more vulnerable to environmental changes or disturbances. Environmental stresses, such as temperature fluctuations, salinity changes, or exposure to toxins, can also influence the evolution of dependencies. Under stress, microbes might become more reliant on helpers for essential functions, strengthening existing dependencies or even driving the evolution of new ones. For instance, a microbe that can tolerate high temperatures might become a helper for a temperature-sensitive species, providing a refuge from thermal stress in exchange for another essential function. This example illustrates how environmental stress can create new opportunities for cooperation and interdependence, reshaping the microbial marketplace. The microbial marketplace is not a static entity. It's a constantly evolving system, where dependencies shift, new interactions emerge, and the balance of power fluctuates in response to environmental pressures and evolutionary innovations. The Black Queen hypothesis provides a framework for understanding this dynamic interplay, highlighting the importance of leaky functions, public goods, and the intricate web of dependencies that connect microbes in a shared environment. It's a story of cooperation and competition, of give and take, where the fate of individual microbes is inextricably linked to the fortunes of the community. By shedding light on the microbial marketplace, the Black Queen hypothesis deepens our understanding of the fundamental principles that govern life on Earth, from the smallest microbes to the largest ecosystems. Chapter 5. The Vanishing Act: How Microbes Master the Art of Outsourcing. The Black Queen hypothesis paints a picture of a microbial world where shedding genes, like a shrewd card player discarding a burdensome hand, becomes a winning strategy. But how does this vanishing act, this seemingly counterintuitive loss of essential functions, actually happen? It's not a random disappearing act, but a carefully choreographed evolutionary dance driven by the relentless pressure of natural selection. A dance where microbes learn to lean on their neighbors for survival. Imagine a microbe swimming in the vast, nutrient-scarce expanse of the open ocean, carrying the genetic blueprint for producing catalase peroxidase, an enzyme crucial for neutralizing the toxic hydrogen peroxide that forms when sunlight interacts with seawater. This enzyme, however, is leaky, meaning some of it inevitably diffuses out of the cell, creating a shared detoxification benefit. Now, imagine a mutant arises, a microbial minimalist that has lost the gene for catalase peroxidase, CATG. In the iron-limited environment of the open ocean, shedding this gene offers a significant advantage. Iron is a precious commodity, essential for catalase peroxidase function, and by losing the gene, the mutant frees itself from the metabolic cost of acquiring and utilizing this scarce resource. It's as if the microbe has outsourced its waste management, relying on the leaky catalase peroxidase produced by its neighbors to keep its immediate surroundings clean. This mutant, unburdened by the cost of enzyme production, can now redirect its resources towards faster replication, outcompeting its enzyme-producing counterparts. This is the essence of adaptive gene loss, a strategic shedding of responsibility driven by the selective advantage it confers. The CATG-deficient mutant becomes a beneficiary, thriving in the peroxide-free environment created by its helper neighbors, while the helpers continue to shoulder the burden of catalase peroxidase production. This helper-beneficiary dynamic isn't limited to peroxide detoxification. Many essential functions, from the synthesis of vital nutrients to the breakdown of complex food sources, can become leaky, creating opportunities for gene loss and the evolution of dependencies. Consider the diffusion of hydrogen peroxide itself. Because it readily passes through cell membranes, any microbe producing catalase peroxidase inadvertently creates a peroxide-free microenvironment, a shared benefit for its neighbors. This leakiness creates the conditions for a Black Queen scenario, where the loss of CATG becomes a viable evolutionary strategy. Gene loss isn't always a clean break, a simple deletion of a gene from the microbial genome. It can be a more nuanced process, a gradual fading of function driven by a variety of mutations. A mutation in the promoter region of a gene, for instance, can reduce or even abolish its expression, creating a cheater phenotype without completely deleting the gene itself. It's a subtle form of sabotage, silencing the gene's instructions without removing the blueprint entirely. This can lead to the emergence of a weak cheater, a microbe that still contributes some public good but benefits disproportionately from the shared pool. Imagine a microbe with a mutated promoter that reduces catalase peroxidase production. It still contributes some enzyme to the environment, but less than its unmutated counterparts, gaining a slight advantage while still benefiting from the overall detoxification effort. Another pathway to gene loss involves mutations that inactivate the protein itself while preserving its production. Imagine a mutation in the CATG gene that renders the catalase peroxidase enzyme non-functional. The microbe still invests resources in producing the protein, but the protein itself is useless. In the presence of helpers, this mutation is near neutral, offering neither a significant advantage nor disadvantage. However, it sets the stage for further mutations that can eventually eliminate the gene entirely, a gradual dismantling of a now useless piece of cellular machinery. Horizontal gene transfer, HGT, the microbial equivalent of swapping playing cards, adds another layer of complexity to the Black Queen dynamic. HGT, the movement of genetic material between unrelated organisms, can both facilitate and reverse gene loss. It can facilitate gene loss by providing an alternative source of a leaky function. If a microbe acquires a new gene through HGT that performs a similar function to an existing leaky gene, it can shed the leaky gene without consequence, relying on the newly acquired gene or the public good provided by others. Conversely, HGT can reverse gene loss by reintroducing a lost gene when environmental conditions change. Imagine our CAD-G deficient Prochlorococcus drifting to a region of the ocean where helpers are scarce. Through HGT, it could reacquire the CAD-G gene from another microbe, regaining its independence and the ability to detoxify hydrogen peroxide on its own. The CAD-G gene itself provides a compelling example of HGT in action. Phylogenetic analyses have revealed that this gene has been shuffled between different cyanobacteria lineages, highlighting the dynamic nature of gene presence and absence in microbial communities. It's a microbial game of genetic hot potato, where genes are passed around, lost, and regained, constantly reshaping the landscape of dependencies. The interplay between gene loss, HGT, and environmental context is a constant push and pull, a dynamic dance where the rules are constantly changing. Environmental factors, such as nutrient availability, the presence of stressors, or the composition of the microbial community, can influence the selective pressures that drive gene loss. In nutrient-poor environments, like the open ocean, the need to conserve resources drives genome streamlining, a process that predisposes microbes to gene loss and the development of dependencies. It's a matter of economic necessity, shedding unnecessary baggage to maximize efficiency in a resource-limited world. The presence of stressors, like toxic compounds or fluctuating temperatures, can also accelerate gene loss. If a leaky function becomes too costly to maintain under stress, microbes might shed the burden, relying on helpers for protection or assistance. The Black Queen hypothesis can even lead to a strong scenario, where all members of a community become interdependent through gene loss. Imagine a closed system, a microbial microcosm, where each species has lost a different essential function, relying on others for the missing piece of the puzzle. This creates a tightly knit community, where the fate of each species is inextricably linked to the others. This interdependence, while promoting efficiency and specialization, can also make the community vulnerable to disturbances. The loss of a single species, or a change in environmental conditions that impacts a key helper, can trigger a cascade of failures, potentially leading to the collapse of the entire community. It's a high-stakes game of interdependence, where the strength of the network becomes its greatest weakness. The Black Queen hypothesis, then, is not just a story of gene loss. It's a story of adaptation, of how microbes learn to navigate the complexities of their environment by relying on the strengths of their neighbors. It's a story of cooperation and competition, of give and take, where the microbial marketplace thrives on a delicate balance of dependencies. It's a story that challenges our traditional understanding of evolution, revealing a hidden layer of interconnectedness in the microbial world, a world where losing, sometimes, is the smartest way to win. Microbial evolution isn't always about gaining complexity. The Black Queen hypothesis reveals how shedding genes, like outsourcing essential functions, can be a winning strategy. Leaky functions create shared resources, fostering interdependence and a microbial marketplace where cooperation and cheating coexist. Environmental pressures, horizontal gene transfer, and the emergence of keystone species shape this intricate network. This understanding of microbial communities has implications for ecosystem stability, resilience, and even human health. What are your thoughts on this evolutionary gambit? Share your questions and ideas below. Like, subscribe, and suggest topics for future explorations. Keep asking questions. The microbial world is full of fascinating secrets waiting to be uncovered.